of you, and welcome back to the third metabolic concept lecture. In this lecture, we will be discussing pyrimidine metabolism, focusing on the de novo synthesis of pyrimidines and their conversion by kinases, and the synthesis of TMP, deoxy-TMP, for the synthesis of DNA in S phase, and finally shedding light on the regulation of the de novo synthesis of pyrimidines. Synthesis of pyrimidines is a simple affair than that of purines. First and foremost is because of it is only one ring structure in pyrimidine, while in the purine it was a double ring structure. In contrast to purines, also pyrimidines are not synthesized as nucleotide derivatives. Instead, they are synthesized as a ring system first, which is completed, and then during the end stages of the de novo synthesis, ribose is attached to the already completed ring system. Seeing the ring system of the pyrimidine, we see that aspartate donates most of the atoms. 4 out of 6 are donated by aspartate, while as the 2 come from the glutamine and carbon dioxide. They come together as carbamyl phosphate. So only 2 precursors are there, carbamyl phosphate and aspartic acid, which contributes to this 6-membered ring. De novo synthesis of pyrimidines like purines is a 3-step affair or 3-phase affair. The first phase is the conversion and production of carbamyl phosphate. Second to 6th step is the phase which involves the synthesis of uridine monophosphate. Then finally, the conversion of uridine monophosphate to thymine monophosphate or cytidine triphosphate, which are the last steps of the pyrimidine biosynthesis. So the whole biosynthesis process up till the synthesis of uridine monophosphate involves one aspartate and then ammonia group of the glutamine, carbon dioxide, water molecule, NADH as the cofactor, and it utilizes ATPs, around 2 ATPs till UMP synthesis. Then there is the utilization of the methyl group for the conversion of uridine monophosphate to deoxythymine monophosphate, or in the second phase where the conversion of the uridine monophosphate involves the conversion to CTP. It involves glutamine which donates the ammonia, and further the utilization of 2 ATPs. This is the whole summary of the pyrimidine biosynthesis. In the current diagram, you will see the 6 steps of the pyrimidine de novo synthesis. It involves the conversion of the carbamyl phosphate to uridine monophosphate in a 5-step manner. So the 2 phases are shown in this figure. First phase of conversion to and production of carbamyl phosphate, and then the conversion of carbamyl phosphate to uridine monophosphate in next 5 steps, involving 2 phases of the pyrimidine de novo biosynthesis. In the first step of the pyrimidine biosynthesis, what happens is that the glutamine and the carbon dioxide in a soluble form react with each other in presence of enzyme which is carbamyl phosphate synthetase 2. This is a cytosolic enzyme, and there is another carbamyl phosphate synthetase which is mitochondrial, which is not involved in the pyrimidine biosynthesis. The CPS2 causes the conversion and the production of the carbamyl phosphate, as you can see in the picture. Furthermore, it is a highly energetic and step which involves the conversion on the basis of the energy which is fed by the hydrolysis of 2 ATPs. So here the production of carbamyl phosphate involves the energy equivalents of 2 ATPs for the synthesis. This is the committed step in the pyrimidine de novo synthesis, especially in mammals, animals. Whereas in bacteria, there is another kind of a CPS, so the committed step in bacteria is not this step. So whenever the regulation has to happen in the mammals, it will be happening here, but in the bacteria, the committed step and the regulatory step is not this step. With the production of carbamyl phosphate by CPS2, the pyrimidine biosynthesis is now moved forward towards the uridine monophosphate pathway, which is the five-step which involves the conversion of carbamyl phosphate finally to the uridine monophosphate. In the first step of this pathway, which is the second step of the whole pyrimidine de novo biosynthesis, is a committed step in bacterial pyrimidine biosynthesis, as I have already told you, because here the regulation in the bacteria happen in this enzyme, which is aspartate transcarbamylase. This transcarbamylation reaction involves the conversion of carbamyl phosphate to now carbamyl aspartate. The phosphate is replaced by the aspartate moiety. So here 80% of the pyrimidine ring is also introduced into the synthesis process. Then there is a step second of the pathway, which involves the conversion of carbamyl aspartate to dihydroorotate. You can see there is a cyclization in this step, and the cyclic product which is formed is called as dihydroorotate. Dihydroorotate is not a true pyrimidine in sense, but if it is oxidized, it will lead to a pyrimidine which is called as orotate, which is the second another step. So here the closure is because of the linkage of the NH2 group with the hydroxyl group of the aspartate, which was just added a step ahead. So in the next step, what happens is the oxidation of dihydroorotate to orotate. This oxidation is brought about by dihydroorotate dehydrogenase, which is an enzyme dependent upon the cofactors. In mammals, which is a quinone, and in bacteria, it is NAD flavoprotein. The orotate which is formed is the true pyrimidine in its essence and its structure. Then this orotate is bound to a ribose 5-phosphate, which is donated by PRPP. So the addition of the ribose is in this fifth step of the pyrimidine biosynthesis, and here the orotidine 5 monophosphate nucleotide is formed. The enzyme is orotate phosphoribosyl transferase. So this is the penultimate step of the synthesis of pyrimidines. Step of the pyrimidine de novo synthesis, the OMP decarboxylase causes the decarboxylation of the OMP and leads it to the formation of uridine monophosphate. The carbon dioxide is released here, and the enzyme is called as ODicase also, or OMP decarboxylase. The pyrimidine metabolism till UMP. Then UMP has to go and get converted into two different moieties or two different nucleotides. So in summary, different enzymes and reactions can be said like this. Two condensation reactions happen which form carbamyl aspartate and then dihydroorotate, which are intramolecular condensations. Then there is also dihydroorotate dehydrogenation, which is a flavin dependent enzyme. It is an intramitochondrial enzyme and has oxidizing powers. For mammals, it is ubiquinone dependent. There is an attachment of base of ribose ring to the OPRT, and the ribose is provided by PRPP, which you have seen in the purine metabolism, how PRPP is synthesized from ribose 5-phosphate by activating it. And OMP or ODKs is the nature's most efficient enzyme, and it enhances the rate of decarboxylation of the ultimate step by a factor of 2 into 10 raised power 23. Actually, in the pyrimidine de novo biosynthesis, the enzymes 1, 2, 3 are on the same chain, and the enzymes in step 5 and 6 are on the same chain. So they are multifactorial and multi-activity enzymes. For the conversion of the uridine which we just synthesized, UMP is there, which is then converted to UDP and UTP. We know that the monophosphates get converted into diphosphates or triphosphates with the help of kinases. We have covered it in the previous lecture. You can see in the previous lecture how they get converted. And then CTP can be also formed from the UTP, which we just convert with the help of a kinases. UTP gets converted to CTP in the second reaction with the help of CTP synthetase enzyme. This is the ATP driven enzyme, and it converts the UTP into the CTP by donating of ammonia group from the glutamine and linking it to the pyrimidine chain. So here the CTP synthetase is not only dependent on the ATP but also from the ammonia, which is donated by glutamine. The glutamine after donation gets converted to glutamate, and ATP gets hydrolyzed to ADP and phosphate. So CTP synthesis is dependent upon two things, ATP and glutamine. Glutamine serves as a donor for the ammonia group. So here we can see the UTP is actually ammonified. Ammonification happens. So for now, we went through the synthesis of the two important pyrimidines, that is uridine and cytidine. However, there is a third pyrimidine which is a function of the DNA structure, without which the DNA structure cannot exist, that is thymidine. Thymidine is synthesized from the uridine in a similar reaction as you might have seen in the previous lecture which I covered, ribonucleotide reductase. These reactions are similar to the synthesis of TMP as well. In TMP deoxytype, the enzyme involved is thymidylate synthetase, which converts the deoxyuridine monophosphate to deoxythymine monophosphate. As you can see in the accompanying figure here, you will see the deoxy type of a uridine is converted to thymidine by the help of an addition of methyl group. This methylation happens which is dependent upon as factor which is called as N5N10 methylene tetrahydrofolate. So thymidylate synthetase will function in conversion only when N5N10 methylene tetrahydrofolate will donate its own methyl group to the uridine, leading to the synthesis of deoxythymidine monophosphate. After donation of this methyl group, it gets converted itself into dihydrofolate. Dihydrofolate is then regenerated back to methylene tetrahydrofolate via tetrahydrofolate. The conversion of dihydrofolate to tetrahydrofolate is dependent on another enzyme, which is dihydrofolate reductase. So there is an interdependency of the two enzymes for the conversion of deoxyuridine monophosphate to deoxythymine monophosphate. Further, these both enzymes are in turn also regulated and inhibited by drugs which can target these enzymes and prevent the conversions. So this is another way of seeing the same reaction. The thymidylate synthetase is able to convert the uridine to thymine, and in turn the N5N10 methylene tetrahydrofolate gets converted to 7,8 dihydrofolate, which is then regenerated to 5,6,7,8 tetrahydrofolate having 4 hydrogen atoms, and this regeneration is dependent on NADPH. NADPH is provided by HMP shunt. So dihydrofolate reductase, while converting 7,8 dihydrofolate to tetrahydrofolate, has to dependent on HMP shunt for its functioning. Then in the another reaction where serine is utilized and glycine is given back, this tetrahydrofolate gets converted into N5N10 methylene tetrahydrofolate, so as it will be able to give its methyl to thymidylate synthetase, and thymidylate synthetase or thymidylate synthase will be able to produce deoxy type of thymidine for the DNA structure during the S phase of the cell cycle. There is another aspect of, as I already told you in the previous slide also, that these two enzymes are the targets of certain drugs. This thymidylate synthase reaction is the target for the drug fluorouracil or fluorodeoxyuridylate, which is an anti-cancer drug, which prevents the thymidylate synthetase from its reaction, and hence the production of TMP is not happening. When TMP is not there, the DNA cannot be synthesized. Similar effect can be held with the help of a methotrexate. Methotrexate blocks the activity of dihydrofolate reductase and hence prevents the regeneration of N5-N10 methylene tetrahydrofolate, and it will achieve the same effect of preventing the conversion of UMP to TMP and hence prevent DNA synthesis. This is the essentials of the anti-cancerous drugs as well as anti-arthritic drugs. As I mentioned earlier, there are two important enzymes which are regulated. However, they are different in mammals and in bacteria. The regulation is at a specific point. In bacteria, the regulation is on ATPase reaction, while as in mammals or animals, the regulation happens at the first step of the de novo synthesis, which is carbamyl phosphate synthetase 2. And UDP and UTP inhibit this enzyme, while as ATP and PRPP activate it. Also, the end products UMP and CMP competitively inhibits the penultimate enzyme, which is OMP deocarboxylase. Or this is pretty much it about the pyrimidine biosynthesis. In the next video, we will be discussing catabolism of purines and pyrimidines. If you like this video, do visit my Amazon page for the number of different kinds of books which I have published. You can use them and order them in both Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.